Okay, it's my great pleasure to introduce John Hardham, our next speaker from Zoetis, who kindly introduced this session this morning. I'd also like to thank, on behalf of our network and my co-director who's here today, Paul Digard, your very kind sponsorship of this event and the meeting and our network as well. So thank you very much, John.
Well, I've got the dubious honor of being the last talk of the day, which means everyone wants to get out of here and go to dinner and go walk around town and explore the sites and have some fun with dinner, drinks, etc. So, in the words of Monty Python, and now for something completely different, I'm not going to be talking about machine learning or neural networks. We'll be talking about coronaviruses from the standpoint of a medical countermeasure producer. So we showed this earlier today, a wide variety of coronaviruses. We've talked about a few of them. From a medical countermeasure producer, we do have vaccines that we've had on the market for many years for a number of these coronaviruses. For the alpha coronaviruses, of course, I have to figure out which one's the pointer here. PEDV, we developed a vaccine for that one back in 2014-15. There are TGE vaccines on the market. We have FIP vaccines on the market and have for many years. We regularly surveil these, obtain isolates, sequence them, see how far away they're getting from our vaccine, and we try to regularly update strains. Beta coronaviruses, we have a number of vaccines in this group as well. Bovine coronavirus is one right here. We could do a MERS virus for camels, but that's a very small, limited market. We'll come back to SARS. So over here in the deltas, we have developed a Delta coronavirus vaccine for pigs, although we haven't marketed that at this point. And then in the gamma coronaviruses, we have vaccines for IBV for chickens, sometimes also cross-protective against turkey coronaviruses in turkeys. So in the veterinary world, we have a pretty decent surveillance network available to continually test these, look for vaccine escapes, and regularly, with a good cadence, update our strains. And then along came SARS-CoV-2. For each of these porcine epidemic virus, for example, limited to pigs. IBV, limited to chickens. SARS-CoV-2 comes along, and it can infect a very, very wide variety of hosts. And it can jump back and forth between those hosts. You know, with humans, a massive amount of surveillance data is available. In fact, many of the veterinary diagnostic labs even turned over to increase capacity for human surveillance. Everything from massive amounts of genomic sequencing data all around the globe, and we've heard a number of talks today about the inordinate amount of sequencing data. The percent sequenced around the globe varies by country, of course, and we heard a bit about that today. The death rates and causes by all sorts of different demographics, age, sex, pretty much anything you can imagine has been striated. And of course, tracking the disease all around the globe. Massive amount of human data. This is an extremely highly surveilled disease. We've tracked the genomic sequence variation over time. This is from the Next Strain website. From the start of the pandemic, we tracked the original Wuhan 1 strain through Alpha, around through Delta, and all the way around through all the different Omicrons. Very highly surveilled, massive amounts of data. And then you get to the veterinary side. In many countries, veterinary diagnostics was not even allowed. It took away from the capacity for human testing. In cases where countries did allow veterinary testing, the amount of red tape a veterinarian had to go through in order to conduct a test was to the point that it would take about 3 to 4 weeks or months to get through the red tape before they could even submit the test. By that point, that animal's either dead or recovered. So there wasn't really a point. So we do have some data. This is from the SARS-ANI website. You can see 34 different animal species, around 39 countries, only about 887 events, under 4,000 reported exposures. Vast amount of underreporting. Some could say 10, 100. We have no idea how much underreported the veterinary disease of SARS-CoV-2 was, simply because we never really opened the door and turned on the light. However, there were some very highly reported cases. We've seen cases in domesticated animals and many in zoologic. There's a number of different newspaper articles. The most highly reported ones were in snow leopards, where there are many deaths of snow leopards around zoos. Surprisingly, even in Florida, they identified manatees as being able to be infected. So this virus can infect a very wide variety of mammalian species, much of which was predicted, some of which was not, based on ACE2 finding and looking at the alignment of the spike protein up against the ACE2 receptor and the strength of the amino acid interactions could make predictions of whether it was likely to be able to bind or not. Some of which we were able to report, like cats, some which we were not, like manatees. And then there's the interplay. I mentioned earlier in the start of this day about the One Health and the interface between animals and humans, as the virus is able to jump from humans to animals back to humans, and the virus changes that are driven in those reservoir hosts that could elicit or open up the growth of subvariants that could then jump back into humans. We've seen a number of cases of this. Very highly publicized case in the Netherlands with the mink variant virus. At one point, this variant virus represented about 20% of the cases in the Netherlands, resulted in the culling of about 17 million mink in that country. There's also been human to zoo animals to human transmission. This was a case right near where I live in the United States of zoo employees that were infected from a lion during a treatment. Of course, there's the case of human to hamsters to human from Hong Kong, where infected hamsters were shipped from the Netherlands to Hong Kong and then infected handlers in Hong Kong, spurring another Delta outbreak in Hong Kong. There's evidence both for and against. I won't say which I believe on this one, but Omicron jumped out of rodent population into humans. There's evidence both for and against that. However, it does infect rodents. And then there was a cat to human transmission that was identified in Thailand. And this was a well-documented paper. And then a very interesting one is what's been seen in the United States is the presence of the virus in deer populations. In this case is a white-tailed deer, in case where human to, in reality it was human to mink to human to deer to human transmission cycle from Michigan into Ontario. And this very interesting paper, this deer variant virus actually had, well, Omicron had 32, the initial Omicrons. The deer variant virus had 76 different mutations away from Wuhan 1. And that was found in humans in Ontario. This is a case I mentioned from the Potawatomi Zoo in Indiana in the United States. A 20-year-old African lion had been vaccinated with the Wuhan 1 based vaccine, which I'll touch on in a little bit. This happened to be during the Delta variant outbreak, was infected with Delta variant. Happy to say that this lion had very minor symptoms, resolved within 5 days, but that lion happened to be having a health treatment at that point. During that treatment, for something unrelated, they found out that the lion did have infection with Delta variant, sneezed on some of the handlers, and then we saw the human handlers, the vet techs, got sick. And then this is from the case in Thailand, where a infected cat from a household was quarantined away from the infected owners, brought to a veterinary clinic for assessment, and happened to infect some of the veterinary technicians. And this was tracked genomically as well. Very interesting case. We know in animals, symptoms of COVID-19 are very similar to what we see in humans. Very similar array of symptoms that have been observed. Just as in humans, we also see an age dependency of symptomologies. We've seen different symptoms based on the variants. The Delta variant was very different in animals than Wuhan 1 was. We also see Omicron's very different in animals. So one of the interesting things was many of the initial susceptibility studies were done with Wuhan 1 in juvenile animals. And then that was used by many regulatory authorities and governments to say these animals should or shouldn't be infectable or able to be infected and may or may not need vaccination. But that was extrapolated to Delta variant and to older animals, which was highly inappropriate. So early on in the outbreak, when the initial cases were reported in Hong Kong of dogs being infected, we started developing a vaccine for veterinary use. We used, instead of the mRNA-based technologies, we used a recombinant protein system. And we expressed in a eukaryotic system the stabilized ectodomain. This was then formulated with a proprietary adjuvant system. You can see the recombinant protein in electron micrographs has this typical cone shape with an elongated tail. And when it's overlaid with the crystal structure, we have an exact match. So a recombinant protein is very similar to the native pre-fusion conformation. We made this vaccine, the experimental vaccine, available to zoos around the world. Initially, we donated about 26,000 doses. It was used in about 225 organizations around the world. You can see the map here. This vaccine was then licensed in the United States for use in mink and was used in about 3 million mink in North America. And we also made it available, again, to zoos, aquariums, conservatories, and other animal organizations. We made a zoologic serial of this and donated about 80,000 doses were made available. And we've had requests from a number of different countries all around the world. And all totaled, about 300 different mammalian species have been vaccinated with this vaccine. That's a first in the history of vaccinology that one vaccine was used in that many different animal species. But one thing this did underscore was the importance of One Health. It's a very striking example of that. We've seen many 30 to 50 different animal species able to be infected. We've seen a number of different cases of transmission from humans to animals and animals back to humans. We've seen reservoirs crop up. And in one study in the United States, surveilling deer, about 80% of the deer were serologically positive in that study. In a more recent one during the Omicron wave, about 20% were actually PCR positive for Omicron. So we have a very large reservoir that could then create viruses, variants that can jump back to humans again. So this is an example of cases where you have humans interacting with domesticated animals, wildlife, potentially even livestock. So this virus can jump back and forth between, yet we're really only surveilling one component of this and leaving the rest of this as a black hole. Now, there are organizations that are interested in funding this sort of research. I just call out one here. This is STAR-IDAS, the International Research Consortium on Animal Health. Their goal is to accelerate delivery of disease control tools and strategies to help with the One Health approach. This is directly off their website. I'll just point out a couple of the research priorities that are in line with what we're talking about today of creating more data around diseases that affect both humans and animals. One of their priorities is coronaviruses, other emerging diseases, what they call foresight. This is the predictive modeling that we're talking about today. And then One Health. So not only looking at humans, but the ability of these viruses to interact with humans, livestock, domesticated animals, and wildlife, so that we can truly see what the real picture is across all those species. Now, right now, what we have is fragmented surveillance. This is just a couple examples of some of the surveillance systems that are available. The OIE, which is now WOA, World Organization for Animal Health, FAO, SARS-ANI. They create some surveillance data, although very underreported and very limited in animals. And this is, of course, for SARS-CoV-2. Just a few examples of human ones, and I show some of the pictures here. These are all fragmented. There's not a single database and network that we can look at to really track in real time the movement of this virus between humans and animals, and where new variants are cropping up in animals that could be at risk to humans. So similar to what I did in the introduction, I asked ChatGPT, what are the benefits of a global integrated biosurveillance system that could help track, report, and predict zoonotic diseases? So it came up with 2, 4, 6, 8, 11 different items of the benefits of such systems. Everything from early detection, rapid response. So the earlier we know of a new variant that's cropping up, that we could use predictive modeling to say, hey, this one could interact with humans, and it's a potential source from this reservoir into humans. Understanding the disease dynamics, what are the most likely reservoirs? What are the things we need to be worried about? The One Health approach, if we control the disease in animals, we can reduce the risk of transmission to humans. That was one, I forget who it was I was talking with earlier, we were talking about MERS. Can you vaccinate camels for MERS and reduce the risk of transmission to humans? Tracking of variants, we've done a good job with that in humans, but not really in animals. Risk mitigation. This could be anything from, hey, if you go out and interact with deer, be on the lookout. You may be able to pick up a variant virus from deer and help us reduce risks and make good risk predictions. Increasing global collaborations, and then the work that we do, trying to catch up to vaccine development. Because we generally make vaccines from what is most current right now, we identify what the consensus sequence is, we start cloning, producing the proteins, scaling up manufacturing, doing all the regulatory studies. Taking that long amount of time, we're almost always behind the curve in terms of variants, especially with coronaviruses. So if we have a better prediction as what's the most likely next wave, we can get ahead of that by cloning those and getting them ready ahead of time. Reducing the economic impact. We've all seen what the economic damage of COVID-19 has been around the world. Increasing the ability to educate the public. And lastly, that holistic ecosystem health. So those are just a few of the examples that would benefit from not only having a fragmented, but moving that towards an integrated surveillance system, generating data not only for human diseases, but also veterinary, and giving us a real one-time, real-time picture of what the disease burden is. And with that, I'll stop and take any questions. Stop on time.
So, any questions from the audience? Please have a think. And I'll start by opening a can of worms, John, and you can either say you can answer the question or not. So, one of the things we noticed with this virus, obviously, is that it infects a very wide range of experimental model animal mammalian species, right? And I guess my first question to you is that when you think about the examples that you've given, they are human to animal transmission because they are effectively companion or zoological animals. Is that right?
In many cases, in many of their wildlife.
So how, in the wildlife, how do you see that transmission occurring?
From humans to, let's say, white-tailed deer,
Yeah.
for example. So there's a lot of questions about how the virus has jumped from humans to deer. I mean, not many people get a chance to walk up to a deer and, you know, rub noses with it. They tend to run off when you get anywhere near them. There's been several studies, especially in New York, in the US, where over time sampling in the deer, they were able to see a human wave, say Delta variant, and then it cropped up in the deer right after. And now you see Omicron, and then it cropped up in the deer. So there's a number of theories as how that jumped from humans to deer. One is, did it go through an intermediary? Say companion animals, dogs and cats. Indoor, outdoor animals, they go outside, they defecate, deer come up and smell that, and other things with it. They can get transmission that way through a secondary route. Hunters in the woods, many hunters actually don't bring a bathroom along with them. They do the same. Tried to say that as politely as possible. There's could be other contact sources that the deer can come into direct contact with. We don't think it's direct human to deer, but it's likely through some other intermediary source, could be fecal, oral, etc. So there are many routes.
So if we imagine a hypothetical scenario, let's take October 2019, for example, given that this virus is so transmissible, why don't we see it in the wild then?
Exactly.
So
yeah, one would think it would have been found in a number of different animals. One would think it would be found in multiple locations, not just cropping up in one spot. There hasn't been examples prior to fall of 2019 of seeing this virus or very close relatives in wild animals around Wuhan or other southern or eastern China.
Right. Okay. So my tangential question is that so when Paul and I and other co-directors were writing this bid to BBSRC and DEFRA, who kindly funded it, one of the things that we pointed out was that in the pandemic at the start, there wasn't a One Health approach.
At the beginning of the pandemic.
And so what would you say, being more on the animal side, if something like this was to occur again? What would be your top 3 priorities? Or knowing what you know about the animal side and then applying it to the human side?
So, my civilian career, I'm on the veterinary side, and my military career, I'm on the human side.
Yeah.
So kind of spanning the two of them, we literally shut down all the veterinary testing and used the capacity for humans, which means we kept the door locked, we kept the lights off, we didn't even really look. So it weakened our understanding of how this virus can move throughout the global population of mammalian species. And we focused on one. Granted, we're human, we look out for ourselves first, but we missed that opportunity. So one with the virus like coronaviruses would be not only to surveil humans, but also animals.
Yeah.
We did start to at least look at that through modeling of the spike protein and ACE2s, and then some laboratory studies. But even those had a lot of shortcomings to them. So, you know, we constantly relearn how to make the wheel. Yeah. We learn it, we forget it, then we have to relearn it. So we keep focusing only on one species and we forget about all the others. So if I had to go back, it would be to look not only at humans, because we found out very quickly mink are an amazing source of growing virus. If we would have taken a more holistic One Health approach, we probably could have predicted that ahead of time. We should also look at not only vaccinating humans, but vaccinating other animals that are susceptible to reduce the risk of reservoirs.
How about like on the vaccine side of things? Because but you and I were talking about this earlier, so it's a leading question, right?
Yes.
But if you worked on animal coronaviruses, you would know that vaccines are good, but they don't
Yes.
necessarily stop transmission.
Yes.
So how do you get those packages of information over to your human side?
Yeah, I give two great examples of this. So when media and politicians were starting talking about the human vaccines, you heard a lot of language about this will stop transmission, it will stop disease, get vaccinated, protect everybody. Well, that's partially true. You know, we've had coronavirus vaccines for decades. We know from a lot of experience in the veterinary world that coronavirus vaccines are not sterilizing. What they do a really good job at is reducing symptoms so that they're manageable, reducing the time period of symptoms, so you're also reducing that window of infectivity, and reducing the viral load, sometimes even below transmission levels. But they'll still be PCR positive. So then when we started having people who are vaccinated, had a little bit of the sniffles, went and got a test, they're PCR positive, the media is jumping around, this is a vaccine failure. And us in the veterinary world, we're looking at it going, yeah, that's exactly what we predicted. These are not sterilizing immunity. Your PCR test can pick up one viral particle in a sample. You're going to have some. If someone's exposed, they'll harbor it for a little while. They might get sick for 3 or 4 days, maybe 5 days, but they won't be in the hospital and dying. So that's one case. Another case, if you look back to H1N1 in 2009, and this is where I get at, we keep relearning the wheel over and over and over again. H1N1 came out. Many countries are grappling trying to get an H1N1 vaccine out for humans. Well, at that point in 2009, we'd had a H1N1 vaccine for over a decade that was licensed. We actually offered, as a company, to provide governments with the cell lines to grow the virus strains, assay reagents, assay protocols, everything. And we're told there's no licensed H1N1 vaccine. We've had it for 10 years. It's for veterinary use. H1N1 came from pigs. We had all the reagents. In fact, we could have shipped liters of high titer neutralizing polyclonal sera that was used for assays to support the manufacture of that product. We could have shipped it overnight. Instead, they had to vaccinate rabbits, generate high levels of neutralizing antibody. Took over 3 months. We could have sent it to them the next day, but the point was, that's veterinary, it's not human. It's the same virus, all you have to do is requalify the serum. So that's a regulatory hurdle, not a technical hurdle. So we keep relearning this. These diseases are zoonotic. They're coming from animals and into humans. We need to keep learning what we've learned in animals over the years and applying it to humans, and not keep forgetting that these viruses come from animals in the first place.
Great. On that sobering thought, John. Paul, did you have anything, or are you all good? Okay, I'd like to end today's session. John, thank you again for you coming, your talk, and also your company sponsorship.